Welcome to the Royal Swedish Academy of Sciences. Välkomna till Kungliga Vetenskapsakademin. Välkomna tillbaka alla ni som var här igår. Idag har Vetenskapsakademin sammanträtt för att fatta beslut om årets Nobelpris i kemi. Jag heter Göran K. Hansson och är akademins ständige sekreterare. Och med mig idag har jag ordföranden i Nobelkommittén för kemi, professor Sara Snogerup Linse, och professor Claes Gustafsson som är ledamot av Nobelkommittén och en av experterna inom prisområdet. I'm Göran Hansen, Secretary General of the Academy, and with me is the chairman of the Nobel Committee for Chemistry, Professor Sara Snogerup Linse, and also Professor Claes Gustafsson, member of the Nobel Committee. I will start by announcing the prize and read the citation. We will hear about the discoveries and the laureates from our experts, and we hope to have one of the laureates with us by phone. And of course you, ladies and gentlemen of the media, are welcome to ask questions either in English or in Swedish. Årets pris handlar om cellens verktygslåda för att reparera DNA. This year's prize is about the cell's toolbox for repairing DNA. Kungliga Vetenskapsakademin har beslutat att 2015 års Nobelpris i kemi ska utdelas gemensamt till Thomas Lindahl, Paul Modrich och Aziz Sanjar för mekanistiska studier av DNA-reparation. The Royal Swedish Academy of Sciences has decided to award the 2015 Nobel Prize in Chemistry jointly to Thomas Lindahl, Paul Modrich and Aziz Sanjar for mechanistic studies of DNA repair. Für mechanistische Untersuchungen betreffend DNA-Reparatur. Pour leur étude de la réparation de l'ADN. Za izledovanje mehanizmov reparaciji DNK. And now, Professor Sara Snogerup Linse, the chair of the Nobel Committee, will give some introductory remarks. Sara, please.
Damages occur to your DNA every day. In fact, right here, right now. If all those errors were left uncorrected, your genetic material would have very little resemblance to the original chromosomes in your very first cell. Life, as we know it today, is totally dependent on DNA repair mechanisms, as have been revealed in molecular detail by this year's chemistry laureates. Their findings have enormous consequences. As just one example, they have led to insights what may go wrong in conditions such as cancer. This is a simplified model of DNA. It covers 22 base pairs. This little piece in reality is only 8 nanometers long, but every cell of you contains some 100 million times more DNA, which covers a distance of 2 meters. It contains the recipes to make all the proteins needed to build you and make you function. Almost every cell of you have almost exactly the same DNA. So there must be mechanisms to maintain the information. If I were to pull out the DNA from a human body and place it in one row, it would cover the distance from the Earth to the Sun and back 250 times. Although it's so much, it's remarkably similar. We have a repair mechanism, but we also have a replication machinery that copies the DNA at each cell division, and this has a remarkable fidelity. Still, like every chemical process, it makes mistakes at random, so there will be errors. But the DNA can also be damaged by other insults like radiation, or spontaneous decay, or chemicals, poisons, for example, as found in cigarette smoke. All those insults cause a variety of structural changes to the DNA molecule. Therefore, we need multiple repair pathways, as will soon be described by my colleague Claes Gustafsson.
Thank you, Sara. Claes, you ready to give us some insights into this year's prize?
I'm ready. So this is our DNA, our genetic material. And in every single cell in our body, we have genetic material that encodes for the creation of a human being. And this information is stored in DNA and is written with a very simple chemical language containing only 4 letters or 4 bases, A, T, G, and C. And changes to this information or damages to the DNA can have very serious consequences. So what are those consequences? Well, for instance, DNA damage and mutations can cause cancer, and they are also seen as an important contributing factors to normal biological aging. How then do these DNA damages occur? How do we get them? Well, it turns out that DNA, in spite of being the carrier of genetic information, actually has limited chemical stability. Our first Nobel Prize laureate, Thomas Lindahl, demonstrated that there are a number of spontaneous chemical processes in our cells that take place all the time and that breaks down the DNA slowly. For instance, just one of these processes is what you have here. Normally in the DNA you have base pairs between C and G, 2 different bases. But sometimes what happens is that the C breaks down and forms a U instead. U is a base or a letter that you would normally not find in the DNA. And there must be some way to get rid of this. Thomas Lindahl calculated that this reaction happens about 200 times in every living cell every day. So over time, we would actually lose all our Cs. Thomas speculated there must be a repair system, and he went out to search for it. And he could indeed find one, which a repair system that we now know as base excision repair. What this system can do is that it can recognize the U in the DNA, it can flip it out and take it away, and then via a set of other reaction, replace the U with the correct C again. This is just one of many types of base repairs that the base excision repair system can take care of. We know today that there are more than hundreds of different lesions to DNA that can be repaired by the base excision repair system, and it guards the information in our genomes. DNA mutation may also arise during DNA replication, which is the process when the genetic material is copied. And when do you need to copy the DNA? Well, that is when a cell is going to divide. You need to take the genetic information in the first cell, and you need to make 2 copies that can go to the 2 daughter cells. And in order to do this, you need to replicate or copy DNA. And what happens then is that the double-stranded DNA is split open like this, and each of the 2 original strands are used as templates to synthesize new companion strands. And during this process, it is extremely important that we recreate exactly the same base pairs that were in the original DNA molecule, like this one, a TA. But on very rare occasions, 1 in a million, there is actually an error introduced. There is a base coming in that shouldn't be there, and we call these incorrect bases mismatches. Our second Nobel Prize laureate, Paul Modrich, he has spent his life trying to figure out how these mismatches, how they are corrected. And he has found a system which we now know as the mismatch repair system. And this repair system contains a number of different proteins. And what these proteins can do is that they can recognize a mismatch. You see this red-blue thing here is supposed to be a mismatch. It can recognize a mismatch. And then it can also recognize which of the two strands that are newly synthesized. Because you just want to correct the strand that's newly synthesized. You don't want to change the old strand. And then you cleave the newly synthesized strand, and you take away the piece of DNA containing the mismatch, and then the DNA is synthesized again, and we get the correct strand. This is a very important mechanism that increase the fidelity of DNA replication with a factor of about 1,000. And it's a very important factor in preserving genetic information in our genomes. Finally, we can also get damage to our DNA due to external sources like UV light, ultraviolet light, present in ordinary sunlight. And one of the things that can happen is that if we have two T's, the letter T's here in the DNA next to each other, if we get ultraviolet light on these T's, sometimes they can actually be a chemical link formed between them like this that destroys the structure of DNA. This has serious consequences both for the structure and function of the DNA, and we cannot tolerate that. Our last Nobel Prize laureate, Aziz Sancar, has investigated how one can take away this type of UV-induced damages in DNA. And he has identified the components and characterized the mechanisms of what we now know as nucleotide excision repair. This repair system recognizes these chemically linked T's, and then it cleaves the DNA on each side of this UV-induced lesion and cuts away the damaged DNA strand like this. After this, we can synthesize a new piece of DNA which is then chemically correct. This type of nucleotide excision repair is not only important for UV-induced damages, but also take away many other types of damages. For example, those that are created by compounds that we get in when we smoke cigarettes. Together, our 3 Nobel Prize laureates this year have explained basic mechanisms, basic cellular mechanisms at the molecular level that help to guard the integrity of our genomes. And for this, the Royal Academy of Sciences, Swedish Academy of Sciences, is extremely proud to present to them the 2015 year Nobel Prize in Chemistry.
Yes. Thank you, Claes. A great Nobel Prize. I hope we now may have... do we have Thomas Lindahl with us on the via phone line from London?
Yes, I am here.
Oh. God morgon igen, Thomas. Tack för
God
att du är
morgon.
med
Jag
oss.
missade tyvärr första hälften av presentationen. Så jag hörde vad som Sancar, men inte om mig, om Modric.
Ja, fint. Maybe we should continue in English out of courtesy to our international journalists.
Yes.
So, who would like to start asking Professor Lindahl a question? Thomas von Heine.
Hej, Thomas von Heine, Sveriges Television. Jag skulle faktiskt vilja göra det på svenska ändå. Du lär få frågor på engelska senare.
Då säger vi så.
Ja, jag undrar bara, jag är väldigt nyfiken nu när det handlar om reparation av DNA. Höra lite spekulation. Vart är det här på väg någonstans? Riktning att vi kan bli så duktiga på att laga så att det blir evigt liv till slut. Vad tror du?
Nej, evigt liv tror jag inte på. Men många DNA-skador kan ju resultera i cancer och andra allvarliga sjukdomar. Så vi vill motverka de skadorna så mycket som möjligt. Och då måste vi först förstå mekanismen för skadorna etableras.
Tack.
More questions? Who would like to continue? Yes. I think it's Per Snaperud. There is a microphone coming to you.
Does this system work the same way in our cells as in the cells of pathogens? And if there is a difference, is there any medication that utilizes this?
The DNA repair mechanisms are universally distributed. All living cells have repair mechanisms, but they are more or less efficient. And we try to identify weak targets in pathogens where they are poor at repair their DNA, and we can perhaps damage them that selectively.
Could you elaborate a little bit about what kind of pathogens that you are working with or considering?
It's too early to say that we can target a specific pathogen this way. Yeah? We just have to understand that many of the drugs we use damage DNA. And so there is a balance here. We have our own repair mechanisms to try to counteract the DNA damage. When we want to selectively damage cells, like cancer cells or infected cells, the repair mechanisms are less than helpful. But without repair mechanisms, we wouldn't be long-lived.
One final thing. It was mentioned that cigarette smoke damages DNA. Do you know how that happens?
Yes, cigarette smoke contains reactive small chemicals. They bind to the DNA and prevent the DNA from being read properly or replicated properly. So they are mutagens. And once they damage DNA, this can result in a number of diseases, including cancer.
Thank you. Do we have more questions? Yes, one over there.
Hej, Matilda Nyberg från Expressen. Jag tänker ur ett svenskt perspektiv, vad betyder det här priset för svensk forskning?
För svensk forskning, jag följer ju svensk forskning och jag är medlem av Vetenskapsakademin och har gjort bedömningar och så vidare för dem. Bakgrunden till min egen forskning kommer ju från Sverige. Jag fick min ursprungliga utbildning i Sverige. Så nu ringer mig telefonen här.
Ville du fråga något mer? Var det svar på frågan?
Vad tror du att det kan betyda för svensk forskning framåt, så att säga, att en svensk prisas på det här sättet?
Vad detta betyder för svensk forskning? Ja, jag hoppas det uppmuntrar svensk forskning genom att visa att detta är grundforskning som började på Karolinska institutet, och det är basen för allting jag har gjort senare. Så att jag är mycket stolt och tacksam över att ha blivit tränad på Karolinska institutet på det viset. Forskning är en internationell verksamhet, så jag har haft sedan dess många medarbetare i världen, inklusive Sverige.
Tack. Vi har en fråga. Kjell Axelsson.
Thank you for giving me the chance, and congratulations to Thomas. I'm Sufjan Axelsson from Channel Radio and Green Post. I just like to ask you that what was your first reaction when you hear the news, and did you expect these news?
No, it was a surprise. I know that over the years I have occasionally been considered for a prize, but so are hundreds of other people. So I feel very lucky and proud to be selected today.
Thank you. Yeah, please.
I'm Per Snaperud från Forskning och framsteg. Could you say something about what led you into this research? Why did you think it was an interesting problem to figure out why Cs get converted to Us and back?
Yes, I was studying the properties of DNA and found, somewhat to my surprise, that DNA is much more labile than we usually recognize. It gets damaged in cells by the water we live in, and that cannot be avoided. So that means you have to repair the DNA all the time. That wasn't known, and nor were the mechanisms known that deal with this spontaneous DNA damage. So we have clarified that. And some of those mechanisms are also used as defense against dangerous compounds in the environment and drugs and so on. But the main purpose is to provide defense against unavoidable DNA damage.
Thank you. Next question.
Hello, David Keaton from the Associated Press. Professor, we're hearing the words cancer, we're hearing the words smoking. These are things that touch. Everyone can understand the impact of these diseases and how they can affect the lives of millions of people. Can you tell us a little bit how this breakthrough in science can lead to better treatment and better medicine?
Yes. As I mentioned, it's very important that we have DNA repair in, unfortunately, individuals that have defective DNA repair. They die early or have serious medical problems. So we have to have DNA repair. On the other hand, if we treat the cancer cell with an anti-cancer drug, that often acts by damaging the DNA of the cancer cell. So we try to kill the cancer cell, and the cancer cell fights back by DNA repair. So we have two edges to this sword. We need the DNA repair, but we don't like it that the cancer cells can repair the DNA. So we have to understand the mechanism so we can selectively provide good therapies that way.
So for the millions of people that are going to be hearing about this prize today, what sort of hope should it bring to them?
In the somewhat longer run, we can provide better treatment and better drugs because we have to understand how DNA is damaged. We can't avoid DNA damage. We live in a world where we get exposed to DNA damaging agents all the time, and many of those are in our cells, and we can't avoid them.
Thank you. Do we have more questions? Pastor Naprud has another one that he needs an answer of. Can we get the microphone?
I'm sorry for so many questions, but it's very interesting. I'm thinking about viruses. Do they have this mechanism, or how are they related anyway to this research?
Yes. Bacteria and large viruses can code for their own DNA repair mechanisms, and that helps them overcome the defense mechanism of the host. That is not always a good thing for us. So we have to understand how the DNA repair mechanism works, and this is what I and others have been doing. So we can, for example, by radiation, selectively kill tumor cells without damaging the individual.
Thank you. Do we have... Yeah, the lady over there. You'll get the microphone right away here. Yeah.
Anneli Megner Arn, TV4. Jag tar det på svenska också. Jag tänkte höra om du kan säga någonting. Jag förstår att det är svårt att säga konkret, men lite grann hur man skulle kunna boosta de här försvarssystemen så att de klarar av cancer till exempel.
Ja, vi vill nog inte klara av cancer. Vad vi vill göra är att selektivt döda cancerceller. En cancercell är en abnorm version av en normal cell. Och vi måste förstå hur de försvarar sig så att vi kan inaktivera cancercellerna utan att skada de celler som i vår egen kropp.
Och hur gör man det?
Genom att ha selektiva behandlingar och sedan många år använder man radioaktiv strålning, kallas radioterapi. Och det är fortfarande ett av de allra viktigaste medel vi har att behandla cancer.
Det här är ju ett av de heta områdena inom cancerforskningen idag, att kunna förhindra att cancercellerna reparerar sitt DNA.
Precis, ja.
Har vi fler frågor här? Alla är nöjda så långt. Då tackar vi Thomas Lindahl för att han var med oss. Thank you very much, Thomas, for being with us today. Congratulations again to your Nobel Prize and welcome back to your old hometown in December.
Thank you very much, and apologize to the English-speaking people who have to sit through the Swedish presentation too.
No problem, I think. Thank you very much. Bye-bye for now. Okay, let's move on to questions to our experts on the panel. Questions from the media. Have we exhausted all questions already, or do you have anything you'd like to bring up, ask us about? This is your chance. Yes, please.
I would just like to know if they have collaborated, the three laureates, or if...
Not to my knowledge. They have worked completely independent of each other. So.
And it's different mechanisms for DNA repair, as you said. Yes, more questions? Okay, yes, Joanna Rose, please.
Yes, I wonder, there are three different mechanisms you just mentioned. Are there many more mechanisms that the research is going on?
Definitely. I mean, new DNA repair mechanisms are being discovered as we speak, probably. So there are many other mechanisms that have been discovered in recent years. And one can say that when we talk about these prizes, we talk about the original discoveries in bacteria often. But what has happened is, of course, that all these three Nobel laureates, they have continued with their work for many, many years, and they have transferred their knowledge into the mammalian system, so they see that the same principles apply also in our human cells. And as I said, there are definitely other forms of repair which are also very important. But these are very early discoveries, and they opened up the field, understanding that mechanisms that this could exist.
When were they made?
So the first discoveries were made by Thomas Lindahl, and that was in the early 70s that he made his discoveries. Then the transformation in understanding how it works also in human cells, that took another 10, 20 years before sort of that come to, I wouldn't say completion because it's still studied. The others did their breakthroughs in the 80s and middle of the 80s, late 80s. That's when they did the breakthroughs. But also Sanjar and Mudrich continued and transferred the knowledge from the E. coli and bacteria into the mammalian system. And then we are into late 90s and early 2000 even. So.
Thank you. Yes, please. Over there. You're getting a microphone.
Just a short question. Why did it take so long for you to give them this prize?
Sara.
Yeah, that's a question that's, of course, is difficult to answer, but each year we have a number. We have close to 400 nominations. So every prize cannot win every year. I guess that's the simplest answer.
I think it's also fair to say that this story has unraveled step by step, and the enormous importance has become clear more and more during more recent years. Please.
As Thomas Lindahl is a member of the Academy, could he have been here today voting as well? Or, I mean, how common is it that you give a prize to a member of the Academy?
Oh, that's very rare indeed.
Has it happened before?
I think so. The last prize in chemistry was in 1948 to Arne Tiselius, and I wasn't even a member then. He was probably a member, but I don't know how it was handled. Thomas Lindahl has not participated at all in this process, not attended any of the meetings or contributed in written form to any of the evaluations, of course. More questions? Yes, we have one over there.
Hello, yeah. Matilda from Nordic Chinese Times. Wondering a little bit from the Chinese side, just about, not about this prize at all, but about the nomination process. Because there's a lot of Chinese people like often wondering how this process is going, like how it's proceeding and how you send out information. Are you actually sending out information to a lot of universities around the world about how to nominate people for this prize?
Yes, this is exactly what happens. So a lot of universities and also individual scientists around the world get a letter where they're asked to nominate.
Okay. Is it also in all the different languages and all the different universities?
The letter is in English,
okay,
but it's sent to universities all over the world. And we pay special attention also to make sure that we include all parts of the world.
All right, thank you.
And we invite nominations from individual scientists at universities. We don't necessarily ask for nominations by the president of the university or the minister for science or anything like that. It's nominations by the scientists themselves, by the international scientific community. And we, as Professor Snugerup-Lindsey said, we really work hard to make sure that universities all over the world are invited to nominate. It could be scientists in China, one university, Peking, 1 year, Shanghai the next, universities in South and North America, Africa, throughout Asia and Europe and Australia. We don't cover Antarctica yet, but once there is a research university in Antarctica, we'll make sure they get the chance to nominate too. More questions? If not, then there will be opportunities for interviews with Nobel Committee members for those of you who have requested it. But this session is about to close. We hope to see you back on Monday for the announcement of the prize in economic sciences. Thank you very much for your interest and your attendance. Thanks. Bye.
Ja, okej. Vi kommer.
Claes Gustafsson, you're a member of Nobel Committee for Chemistry.
Yes.
Can you tell us some words about this year's prize?
So the prize this year is about the cellular systems that preserve the genetic information. So it turns out that the DNA molecule, which encodes the genetic information in our cells, that molecule is frequently damaged, and it might also be mutated. And that can be caused by both by external agents like sunlight, the UV radiation in sunlight, by smoking, different agents in smoking. But it can also be internal sources like reactions with water, which happens at a quite high frequency actually. We also get mutations in the DNA when we replicate the DNA. So every time a cell is divided, we need to create 2 new copies of the DNA. And together these different sort of sources of mutations and damage can create a real problem for our genomes and preservation of genetic material. And what these 3 prize laureates this year have done is that they have investigated and described in chemical detail important processes, repair pathways that can correct these mistakes when they arise. And it's both there are 3 different areas of DNA repair that we've been focusing on. One is base excision repair that take care of mutations or chemical damages, I should say, to bases, many of them spontaneous or hydrolytic. We also have the mismatch repair system, which takes care of the rare mistakes that the DNA polymerase leaves behind when it replicates the DNA. And then we have the nucleotide excision repair system, which takes care of, for example, UV-induced damage and other types of, for example, agents in smoking. So that's what this year's prize is about.
Was this a discovery they made, that there is a repair system?
The existence of some sort of DNA repair is something that goes back a very long time. And probably the first identification of a repair system was the discovery of an activity called photolyase in bacteria, which could overcome UV-induced damages. But at that time, the mechanisms for how that happened was not known at all. This was back in the 40s, and then in the middle of the 50s when they saw it in extracts. Actually, transforming those phenomena in sort of cells and in extracts is something that all these 3 sort of laureates have done. They have taken sort of preliminary observations and made it into sort of real chemistry. And when it comes to the photolyase, which I didn't talk about during this press conference, it's actually Aziz Sanjar who has both cloned the gene, identified the protein, and described the mechanism for this really important enzyme. The reason why I didn't talk about this repair today was that this is actually something that's found only in bacteria. We don't have this type of repair in our cells. But all these other 3 types of repair, they were first identified in simple cells, and then it turned out that this knowledge was applicable also to our higher human cells, and that we have very similar systems that take care of damage to our DNA.
But the repair system is not enough. We still have diseases caused by the damage of DNA. How big part of the diseases is has their origin in this process?
So one can say that DNA mutations, how to put this? I mean, if you think about cancer, I would say that all forms of cancers start with a mutation or a DNA damage. That's how they start. We know, so I mean, that's how important this is. And if you don't have repair system that take care of the damages as they occur, the frequency of cancer, for example, becomes much, much higher. And there are diseases where you have a disturbed repair mechanisms. They don't work as they should. They are themselves mutated. And then you get very high levels of cancer. One example is scleroderma pigmentosum, a disease characterized very carefully by an excellent American scientist. His name is John Cleaver. And he found all these different genes that cause scleroderma pigmentosum. And then it turns out that all these genes actually, most of them actually encode components of the nucleotide excision repair system in human cells. And the people or the patients suffering from this disease, they cannot repair UV-induced damage as they should, and they get extremely sensitive to sunlight. They need to stay inside, and when they go inside, when they are subjected to UV light, they develop different types of tumors, and they often die from malignant melanoma or this type of skin tumors. So that's the importance of it. But then, of course, a certain level of mutations is also something that's probably needed. So even if the cells try to sort of lower the levels as much as possible, of course, you don't want a frozen genome, because Darwin evolution requires that there are at least some mutations that take place and that we can utilize for selection.
It has to be a little flexible.
Yes, even if I think that the focus in lowering mutation rates, but I mean, they can have positive effects.
Are there any concrete drugs or treatments that have developed from this research?
One can say that we understand now how some treatments work. So, for instance, radiation is something that it's widely used in the treatment of different types of cancers. And one important thing that radiation does is that it induces breaks in DNA. So it breaks down the DNA in cancer cells. And breaking down the DNA means that we overwhelm the repair system. The repair system, there are so many DNA damages that it cannot cope. And then the cells might choose to go into programmed cell death or apoptosis. And so suddenly we understand at least one way that radiation works. There are also new drugs being developed where one tries to utilize the fact that many cancer cells have a defect repair system already to begin with. So by inhibiting repair actually in the cancer cells, you might get something that we call synthetic lethality, and that will sort of specifically kill the cancer cells. This is a very interesting concept that's been currently developed, and I think that there are a number of different pharmaceutical industries that are looking into this, and I think there are 1 or 2 drugs that are actually already available that builds on this concept.
So this is a field that's very active still.
It's very active, and if you think about DNA damage, I mean, think about all the things that we read about cigarette smoke, we read about avoiding sort of sunlight, or think about catastrophes like that in Fukushima with the nuclear, sort of with radiation. The threat is to our DNA, it's our genomes that are being threatened, and these are the machineries that are trying to fix it for us.
Actually, I thought that Alfred Nobel was a chemist. Chemistry was his field. What do you think he would say about this year's prize?
I think he would be happy and proud. I am very happy. I think that this is a prize in the tradition from the discovery of the DNA, understanding how the DNA is sort of read by transcription, and now understanding how you actually preserve this genetic information, which is so important for life.
That's great. Thank you very much, Claes Gustafsson.
Thank you very much.